Hello students, namaskara. Am I audible, visible? Are we live? I hope the issue is fixed and we are live, right? Yes, we are live. Awesome, awesome. Yes, we are live. Hello. How are you all doing? Yes. Students, let others also join because you know many of them went back and they will join now. Okay, it's almost 7:45 anyways. Yes. So I hope all of you are doing well. Welcome all of you to We Karnataka, KCET and boards. Thank you so much for joining. Hello Charan, hello Sri Vishnu, hello Vedika, Shivani, Babu, very nice names, okay, beautiful names all of you have. Yes, are we ready? So today we are going to do sexual reproduction in flowering plants. Like I always say, being a zoology teacher, I always take up this, chapter, right? So sexual reproduction in flowering plants is a very important chapter, and from KCET point of view, ma'am, what is the weightage, ma'am? How much questions is it going to come? Thank you. Yeah. How much questions is it going to come? Students, you can expect 4 to 5 questions in your KCET, and in some places you will also see that up to 6 questions have come from this chapter. So can you skip this chapter? Definitely not, right? Sexual reproduction in flowering plants is a very important chapter that way, and I have picked the most, questions keep coming from these sections. So I've taken that. What are they? Microsporogenesis, Megasporogenesis, double fertilization. So I'll give you an introduction to it. Don't worry. It's an introduction, then a continuation. Okay. Put a lot of fire emojis in the chat box and put a lot of likes in the, like the video and share the video with all your friends. Okay. I want to see all of your excitement. So you have to put which emoji? Fire emojis in the chat box. Very nice. Okay. Yes, let's start the class. So students, before I go into microsporogenesis, we all know about flowers, right? We have given flowers to our loved ones. We have given flowers to our parents. Parents have given it to us. We have given it to our relatives. So flower is somewhere like a way to express our emotions, be it love, be it sad, be it happy, be it, to congratulate someone for their wedding, congratulate someone for their success. We always give flowers. Now, what is the indication? Why are we giving flowers? Right? We are giving flowers to give or to show our emotions. But is that the only job of flower? Like flower is created so that Aditi and Shivani can give it to her friends? Not really. Flower is actually made by your angiosperm, right? Angiosperms are forming these flowers as a part of their sexual reproduction, right? So that we can form seed out of it, we can form fruit out of it. So this is a typical flower, and what is the ultimate goal or what is the destination that we are going to reach up to? That is, ma'am, ovules are going to become seeds. Okay, are going to become seeds, and the fruits are formed from the ovary. Ovary is going to form the fruits. Yes or no? So this is the ultimate, right? And we consume the fruit, right? We also consume the fruit. So this is the whole purpose of this angiosperms forming your flowers, right? Angiosperms are forming this flower as a part of their sexual reproduction. Got it to all of you? Now look at the flower. Ma'am, I have seen a flower, okay? I have seen a flower and I know that this is how a flower is going to be, right? What and all is there in the flower? First of all, the most prominent thing that you see in the flower is what? The petals, the beautiful petals that you see, right? So these are your petals, and this one, stigma style ovary, this part of it becomes the female reproductive part. And what you're seeing here, which I'm highlighting here with pink, this is your anther that forms, many anthers together form your stamen, right? Clear to all of you? Very nice. Okay, all the energy is very nice. Oh, Charan's energy is infectious. Okay, Charan has sent so many fire emojis. Now students, in the flower, remember, Okay, as you can see, these are your petals. Yes, ma'am, understood. These are my petals. Okay, I will mark it with a different color. These are my petals. Okay, these are my filaments and anther together forming the stamens. Okay, understood. Stigma, style and ovary together forming my carpel or pistil. Okay, now the portion where you hold the flower, no, you hold the flower and give it to someone, no, that is what your pedicel is coming, and above that you have a swollen portion called as your thalamus. Okay, so here you can see, now inside this ovary your ovule is going to be there, and this ovule is going to form the seed, right? This ovule is going to form the seed. So basic is clear? Basic is clear, yes or no? Students, please talk to me, okay, because if you talk to me only, I will know that if my speed is okay, are you able to understand? So keep putting thumbs up when you're understanding the concept, and when I'm asking you, are you understanding? So this way what happened, we will have a communication, right? And we'll become more like friends, okay? Friends, teacher, student, that connection is always going to be there, okay? Very nice. So students, this is clear. Now I will tell you a very important part from your board point of view, and especially from your KCET point of view. In the entire flower, okay, if I want to divide the entire flower, I will divide the flower parts into two categories. First is called the essential, okay? First is called the essential, and second one is called the non-essential. Now what is essential and non-essential? Let's say when you're going to, you know, some supermarket, your mom is writing that essential parts, okay? That is Charan or Babu, when you're going to supermarket, you are not supposed to buy chocolates, you know, Fanta, Sprite, Coca-Cola. What are you supposed to buy? You have to buy half kg rice, dal. So that means what? The most essential things for your home is what your mom is trying to talk, right? So in case Charan and Babu does not bring essential parts, he'll become non-essential in the house, right? Your mom is going to throw them out of the house. So same thing here, essential and non-essential means the most essential part of the sexual reproduction is going to be two people, students. Who are they? Your andrecium and gynecium. Clear? Gynecium. These are your essential parts. Now andrecium means what? The male part, okay, and gynecium means the female part. So you can remember GG, okay, gynecium, the female part, okay. So andrecium and gynecium. Now ma'am, what is this non-essential parts? See, even though we know that rice is necessary, dal is necessary, there are some things that we buy, you know, like maybe some chips packet, some apla, or you know, papad, something like that. We buy other things. They are non-essential, they are not something that you will consume directly, but it adds a taste to your food, right? It adds a taste to your food. So same way, your non-essential parts are going to be, students, the ones which are going to help for the sexual reproduction, like your petals. So I'm going to write the words, okay, corolla and your calyx or calyx, okay. Now ma'am, you have written andrecium, gynecium, corolla, calyx, but you did not write anything about the other terms that you spoke about. Students, who is going to be part of the andrecium? Since I am telling that it is the male part, stamen is going to be a part of it, right? And stamen consists of what? Anther and filament, as you can see here, okay? Now ma'am, gynecium, who is going to be there? Your stigma, style and ovary, which is going to form the carpel, and carpel is going to come under, many carpels together will form your gynecium. Clear to all of you? Yes? Yes, very nice, very nice, okay. Non-essential part means, ma'am, you have written corolla. I've never heard about corolla. Who is going to come under corolla? Your petals are going to come under corolla, right? Petals are going to be a part of corolla. Then what about calyx, ma'am, or calyx? That is going to be, students, your sepal. Sepal is going to, many sepals together, the units, together it is going to form your calyx. Is this clear? Yes or no? Yes? Clear? Thumbs up. This is clear, essential and non-essential part, because this can be asked in your KCET. Which is your essential part, which is your non-essential part. Why is it called the essential part, right? It is called the essential part because they are directly helping in reproduction, right? It is forming the male gamete and the female gamete. But what about the non-essential part? They are helping in attracting the insects, because if the pollen grains are not being transferred from one flower to another flower, cross-pollination will not happen. If self-pollination continuously keeps happening, then inbreeding depression will happen. Can you imagine? Can you imagine? So everything is so aligned, okay? Now, we are going to understand the main portion, that is your stamen. If you understand stamen only, I'll be able to explain you microsporogenesis. If you understand carpel, I'll be able to explain you megasporogenesis, okay? Very nice, okay? All of your energy is beautiful. I hope all of you are liking the video as well. Don't wait for me to tell, okay? Don't wait for me to tell. Yes, very nice. Like the video quickly, quickly. Let's make 50 likes as soon as possible, okay? And share the video with your college friends. Now, students, stamen, as I told you, the unit of your stamen is going to be your anther. Like, what is your entire stamen going to have? This is your filament, this is your, okay, anther, right? So, here, this portion is going to be called the filament. Have you all seen this? Okay, filament. And this bilobe structure is going to be called your anther. This together is going to form your stamens, right? And many stamens together is going to form your whorl, which is called as your andrecium, which is the essential part. Ma'am, all this is clear to me. So, students, as you can see from the flower, one anther is taken. Now, one more very important statement is given. Yes. Mala Pushpa, yeah, Pushpa, you can use this, okay? You can watch the video, don't even worry. Yes, now, students, let me tell you one more important thing. What is the important thing? That when I say this filament, see, the filament can be attached to a petal. Now, let's take the filament is attached to the thalamus, right? The swollen part. The swollen part which I spoke about is your thalamus. Okay, this is your thalamus which I am attaching your filament to. Now, there is two words which is mentioned, and maybe in your KCET it can be asked in the MCQ part. That is, what is in the proximal end of the thalamus and what is in the distal end of the thalamus. Now, all of you tell me, proximal means what? Proximity. There is close proximity between a NEET aspirant and a KCET aspirant, because both of them are preparing for a competitive exam, right? One is for an engineering seat or a B Pharma seat, one is for a medical MBBS seat. So, close proximity means what? Both of you have something very similar, that both of you are attempting for the competitive exam. Distal end means what? Your distant cousin, we tell, no? That means the person who is far from us. So, this portion, right? This portion is going to be, so filament is at the proximal end of the thalamus and your anther is at the distal end of the thalamus. Is this clear to all of you? There is one line given in your NCERT and questions can come from it. Yes? Fine. Okay, fine. Okay, this is your proximal and distal end. This stamen part is clear. So, anther is there, filament is there. Now, what you are supposed to know about each of them is very important. Now, let's say I cut, okay, I take a cross section of this, like this. Okay, I do like this. I'm going to get what? I'm going to get a structure like this, right? I'm going to get a structure like this. Okay, I've cut this portion, same thing like this I'm going to get, okay, like this. Now, if I want to see the inside of it, okay, if I want to see the inside of it, I have to check, right? For that, let's say in your NCERT, you will be able to see a cross section which looks like this. Yes, like this one diagram is given in your NCERT or not? This diagram is given, right? Yes. So, inside the students, they have drawn globular structures like this, right? In the corners. What are these? What are these? So, I have taken a cross section, okay, I have taken a cross section of anther. These are my two lobes, okay. In this lobe, 2 theca is there, in this lobe, 2 theca is there. Is this clear to all of you? Okay, now, look here, there are 4 globes here, okay, 4 globular structures. Now, I am going to explain you everything. Now, students, 2 anther, right, this is your 2 lobes of the anther. Let's say this one hand of mine, where this ring is there, this is one lobe, and where this ring is there, this is one lobe, okay. So, this one lobe, they are attached like this with the help of a connective. With the help of a what? Connective, okay. Where is that connective going to come? Ma'am, obviously connective is going to come here. Clear to all of you? Yes. So, we will label this is going to be your connective, okay, connective. Now, this is one lobe, this is one lobe, and you will see these 4 structures, okay, we call them theca. What do we call them? Theca, okay, theca. So, I can say that in one lobe there are 2 thecas. So, can I call my, anther as dithecus? Yes, ma'am, you can call anther as dithecus, right? Can I call it dithecus, students? Because in one lobe there are 2 thecas. So, can I call, in each lobe it is going to be dithecus. Yes? Oh, all are telling hi to each other. Yes, clear or not? Okay. Ma'am, can you say answer for an order of male gametophyte? Yes. I'm going to write that, Monica. So, that doubt of yours will be clear, okay? Yes. So, dithecus. Understood, ma'am. Now, look at this. Inside this, students, inside this, there are going to be, layers to this, okay, layers, which we are going to learn later. Now, look at this. So, you understood that your anther, this anther is actually a bilobed structure. What is your anther? A bilobed structure, because this is one lobe, this is another lobe, okay. They are bilobed. Bilobed, dithecous, and how many microsporangium? These are microsporangium, okay. Inside this only your pollen grains will be formed. So once this mature, I can call it the pollen sac, okay. I can call it the pollen sac. So look here, 1, 2, 3, 4. So can I call it tetrasporangiate? Yes, ma'am, you can call it tetrasporangiate because there are 4 to it, okay. There are 4 to it. So tetrasporangiate. Okay, tetrasporangiate. Now students, look at this diagram. All of you can see the diagram as well. Yes. Oh, what is happening? Okay, look here. Students, here anther is there, right? Anther is there. And this anther, if I cut it, you can see how they are. And inside this, can you see these circles? Can you see these circles? These are your microsporangium. Okay, microsporangia. This microsporangia has to mature. Okay, this is going to mature, and after they mature, I can call it a pollen sac. I can call it what? A pollen sac. Now, remember, in the center of this microsporangium, there are going to be a set of cells. Okay, there are going to be a set of cells which are called as your sporogenous tissues. These tissues are capable of becoming your pollen mother cell or your microspore mother cell, okay. This center portion will be filled with the sporogenous tissue. Yes, sporogenous tissue. This is your sporogenous tissue. Is there anyone who is studying this chapter for the first time? Anyone who is studying this chapter for the first time in your life? Oh my God, first time I am opening this chapter. No one, right? No one should be there like that, okay? Yes. Yes, Tanuja is taking the attendance. I want all of you to do the attendance also. So, sporogenous tissue is clear. Now, we will go to Monica's doubt about that, okay? But before that, we have some more layers to study. So, students, this is what I drew, okay? In that, we drew the theca. So, we know that anther, as of now, we know few things about the anther. We know that anther is bilobed. We know that the anther is bilobed. We know that the anther is tetrasporangiate. Tetrasporangiate. Okay, then we know that the anther is dithecous. Okay, and according to the shape also, people tell it is tetragonal in shape. Like, how does it happen, ma'am? Tetragonal in shape. Like, let's say I take this, okay, I do this too, okay, like this, okay, like this. Okay, tetragonal in shape. Got it? So, according to my marking, you can see, no, tetragonal in shape. Okay. So, that, sorry, yeah. So, that is your tetragonal in shape. So, somewhere if you see that, don't get it confused. Understand that it is, they are talking about the shape. Okay. Now, I told you this microsporangium has layers to it. So, imagine students, you have diamond. Okay. You have diamond in your home. Okay. One big gem you have. Will you just keep it outside like this? But you will not tell like that, no, you will keep it inside, you will pack it and keep it. Maybe your mom will keep it inside the rice also with a cloth covering. How do we keep it? So imagine if someone is capable of forming the male gamete in the future, don't we protect them? We have to protect them, right? We have to protect them with as many layers as possible. So same thing is going to happen here. There are many layers to it. Okay, for protection. So, in between there is sporogenous tissue, but until we reach the sporogenous tissue, there is many layers of covering, right? All of you tell me, if you have a diamond in your home, how are you going to protect it? You're going to protect it with many layers, right? Many layers as possible, no? So, like that. Now, many of you would have kept your biology book like that, you know, covered and kept it newly, okay? Like, when I sell it to the next person, the person should doubt if it is a new book or it is a book used by my senior. Right, all of you are keeping your book that neatly, I believe, no? Packed it, kept it, dusty it is, don't touch. Okay, sometimes open and see, students. Rat or cockroach or frog or something might be living inside, so open your biology book, all of you, right now. Okay, so look here, these are the different layers. Okay, these are the different layers. So your first layer of your microsporangia is going to be epidermis. Okay, what is it? Epidermis. Even we have now on the skin, epidermis. Okay, the next layer is going to be endothelium. Next layer is going to be what? Endothelium. The third layer is going to be middle layer. Now remember that your epidermis and endothelium are single people. Okay, single people. That is single layered they are. Single layered, but middle layer will have multi layers. Okay, they'll have more layers. That's why when you see the labeling in your textbook also, you will see that it is labeled multiple times, right? 2, 3. Okay. Okay, it's like old book, then I'm happy. It should be like old book. Okay, if it is old book, torn pieces of paper is everywhere, no problem. I'm totally trusting you. But if it is not opened, okay, then you have to open and see. Okay. Yes, fine. Now, next students, as I told you, what sporogenous tissue is there in the midfield, no? But then there is a very important layer after middle layer which is called the tapetum. Now, about tapetum, you have to learn a lot about tapetum. Not a lot, but a little about tapetum. Tapetum is the layer that is actually going to do. See, these 3 layers, epidermis, endothelium and your middle layer is going to protect, and they are also going to help in dehiscence. Dehiscence means what? When anther, see, pollen grains is inside. Okay, now let's say you want to go for a party and you're not wearing that necklace. Okay, not uninucleated, yes, single layer they are. Okay, uninucleated, multinucleated, we will talk about it later, but you do not have to know that. Okay, but remember that tapetum is binucleated or multinucleated. That means they have more than one nucleus. Clear? More than one nucleus. Okay, now remember students, this epidermis, endothelium, middle layer, all the 3 layers are going to do the job of protection. Okay, protection and dehiscence. Dehiscence means what? I have to break through it, no? So the pollen grains can get scattered. If I don't break the anther, how will the pollen grains get dehiscent? Okay, that means scattered. So that's why protection and dehiscence will be done by your epidermis, endothelium and middle layer. Ma'am, then what is the use of tapetum? Tapetum is going to do, definitely it is going to do the protection, students, but mainly it is going to do the nourishment. Mainly what it is going to do? The nourishment. Nourishment for what? For the growing pollen grains. Nourishment for the growing male gamete. Clear to all of you? Clear? Yes or no? My God, what are you people telling? They're telling, when I open the textbook, sometimes I see mosquitoes, dead mosquitoes it seems. Okay? That nicely my students keep the book. Those who haven't subscribed, what are you waiting for? Huh? Which blue moon are you waiting for? Subscribe to the channel, all of you, quickly. Subscribe and like. Okay? This is 2 things my students doesn't do wherever I go. Kanjoos. 1 number kanjoos. Okay. Now students, this is clear. Tapetum is going to do the nourishment. Now, 1 layer is over, epidermis is over, endothelium is over, middle layer is over. Then came tapetum. Inside, between, what is there? Your sporogenous tissue, which is going to, okay, which has the potential to become the microspore mother cell. So when we write, okay, one second, yeah. So when we write here, students, we are going to enter into a microsporogenesis, okay, microsporogenesis. So Monica's doubt was, ma'am, what is the order? Right, what is the order? So the order is going to be like this: sporogenous tissue, okay, or directly you can write MMC and your PMC, no problem. What is MMC and PMC? MMC stands for microspore mother cell. What does it stands for? Microspore mother cell. Okay, microspore mother cell. Okay, meiosis will happen, it will form a diploid, MMC, okay, microspore mother cell, or I can also write PMC, that is your pollen mother cell. Okay, what is it? Pollen. Students, all these notes you will get it on V Karnataka, KCET and both's telegram. Okay, and if you want to join my telegram, it's biology by Gopika. Okay, you can see it on the corner. Yes, clear? Okay, nice. Okay, pollen mother cell. Clear to all of you? So, from the sporogenous tissue, MMC happened. Now, look here, look to the diagram down. I've kept the diagram for all of you. So this is your pollen mother cell, which is PMC. Now what are we doing? We are doing microsporogenesis. That means formation of microspores from microspore mother cells is called as microsporogenesis. All of you repeat after me. Formation of microspore from microspore mother cell or pollen mother cell is called as microsporogenesis. Why are we doing this? We are doing this so that we can form the male gamete. Okay, and now I'll tell you the difference between male gametophyte, male gamete. Male gametophyte is going to have the male gamete. Do not write, say, let's say one bag is there, inside the bag, okay, you have a chocolate. So will you call the bag itself a chocolate? If someone asks you a chocolate, will you give the bag? No, you will give the chocolate inside the bag, no? So the bag is going to be the gametophyte. Inside that, your male gamete is going to be there. Is this clear to all of you? Gametophyte and gamete, because in when KCET exam happened, they will not think that, oh, the student studied gametophyte or gamete. They will think that every student knows this because you are supposed to technically know this, okay? That doubt is clear, no? The bag and the chocolate. So, understand, ultimate goal is that chocolate, okay, has to reach to another person. So, bag is going to be the gametophyte. Inside the chocolate is going to be the male gamete, okay? Gametophyte, gamete. Now, look here. MMC happened. The first meiosis one is going to happen, that is from PMC or MMC. I am writing MMC also. Many students will get confused, that's why. Okay, your microspore mother cells will undergo meiosis 1. Okay, meiosis 1. And that meiosis 1 will lead to the formation of your dyad. Okay, dyad means 2 cells, can you see? 2 cells, can you see? Right? Then it will undergo meiosis 2. Second reductional division, okay, meiosis 2. That meiosis 2 will lead to the formation of your microspore tetrad. Okay, that will lead to the formation of microspore tetrad. This microspore tetrad is like, I don't want to leave each other, okay, I don't want to leave each other, let's all be friends. Now, all of you will become friends, no? Now, by attending your KCET channel, all of you will become friends. You'll be like, okay, whenever you're going for Gopika Ma'am's live, you message me also, even I'll come. So, that becomes a group. Same way here also, four microspores came together, and it is because of a covering as well, but luckily you do not have to know it. Okay, so microspore, for NEET students we usually teach, okay. These microspores together form the microspore tetrad. Now, each of them can become potentially a pollen grain. That means, if let's say, this is your one microspore, this is another microspore, okay, microspore, all of this is an individual microspore, right? Yes? Oh, no, Sagar, you come back when you are in 10th standard, okay? You take a 6 years break and come back, okay? That time I will teach you again. So, students, each of this microspore, okay, each of this microspore can become a pollen grain. So, let's say I have taken this one, okay, this one fellow I have taken and I am going to make him the pollen grain, okay. So, this is your pollen grain. Is this clear? Now, you tell me, is pollen grain the gametophyte or the gamete? Now, let's see how many of you understood. All of you nodded, no? When I told this, all of you understood, you told. Now, tell me, is pollen grain the gamete or the gametophyte? See, each of them will become a pollen grain. Question, quickly answer the question. So, students, let's go once again. Microsporogenesis is where the formation of microspores from the microspore mother cells with the help of meiosis. Okay, here now meiosis 1, meiosis 2 is happening, and that microspore tetrad, each of them can become potentially the pollen grain. Now, tell me the question. Pollen grain is a gametophyte or a gamete? Okay, pollen grain is a gametophyte or a gamete? Question to all of you. If you answer this, you're going to get 1 mark in your KCET. Okay? Yes. Let's see who is answering. Yes, very good, Kush. Very right. Pollen grain is a gametophyte, because inside this there is vegetative cell and generative cell. In the generative cell, the male gametes are going to be there. So, it is a male gametophyte. Okay, very good, Aditya, very good. Okay, only Kush and Aditya has answered, others all have got confused. Okay, so this is why you need to have the clarity in depth. Students, just by reading, it's not biology. Biology has a lot in depth. Okay, lot in depth. So, remember that when you're trying to understand something, you better understand it from a person who is trying to explain you in the simple language. Now, I told you about the bag and the chocolate. So, imagine like that only. This is the bag. Okay, this is the bag. Inside that, there is generative cell. The generator cell is going to have the chocolate. The generator cell is going to have the male gamete. So what is it going to be? Yes, very good Tanuja, I can see your message. Okay. So pollen grain is a gametophyte. Clear to all of you, those who made mistake? Is this clear to all of you? Gametophyte. Okay. And in this you have vegetative cell and generator cell. In the generator cell is going to form the male gamete. Okay. Clarity everyone got? Kushi, Babu, okay, Monica, is this doubt clear? Okay, I really like the name Monica because of friends, Monica, Rachel, okay, it's such a nice name. Yes, this is clear? This is your microsporogenesis. That means from the sporogenous tissue you have your MMC or PMC. This PMC and MMC is going to end up forming your microspore tetrad. Okay, microspore tetrad. You can directly write microspore tetrad also, no problem. Just write meiosis 1 and meiosis 2. Okay, tetrad, and then this tetrads, each of them is going to form a pollen grain. Inside this pollen grain there is generator cell and that is going to form the male gamete. Okay, male gamete. Done? Yes, okay, very nice. Crystal clear? Very nice. Okay, now see students, this is what I was talking about. Okay, this is what I was talking about. That is, see, your microsporangium is clear, no? Now this is what your pollen grains. All of you know hibiscus plant, no? Hibiscus, China rose, we tell. Now inside that, this is your pollen grain. So see how the formation happened. From your microspore tetrad, you formed a pollen grain which looks like this, right? Which looks like this. This is your pollen grain. Right, this is your pollen grain. Now I told you that there is generative cell and vegetative cell. This is the diagram given in your NCERT. Now you have to know the diagram exactly. Okay. Now if you see outside, I have drawn a very design full structure. What is that? This is your exine. Outside layer, outermost layer is your exine, and this exine is made up of sporopollenin. What is it made up of? Sporopollenin, which is one of the hardest, okay, most resistant substance. You pour acid on it, you put alkali on it, you put temperature on it, it will not even move. It will be like, go that side, okay, like that. Sporopollenin is so hard, so resistant to everything, okay. Then the next layer which you are seeing here, okay, next layer, this one is your intine, okay, this is your intine. Intine is made up of cellulose and pectin, and let me tell you, this could be an MCQ question, okay, cellulose plus pectin. This could be an MCQ question, this could be an MCQ question. What is exine made up of? What is intine made up of? Okay. Now, let's get into the inside part. The large cell that you are seeing is the vegetative cell. The large cell that you are seeing is the vegetative cell. Ma'am, what is vegetative cell going to do? It is going to provide the nourishment. It is going to provide the nourishment for the generative cell. Clear? It is going to give the nourishment for the generative cell. Why? Because generative cell has to make the male gamete. Okay, male gamete. Okay. Now this is going to be, this Kuti cell is going to be your generative cell. And in the generative cell, what is going to happen, students? This is going to form the male gamete. So now you tell me, is pollen grain a male gametophyte or a male gamete? All of you tell me this. Is it a male gamete or is it a male gametophyte? Yes, this is clear. Yes, it's a Kuti cell, no? Now, look here, this is the diagram given in your NCERT. You can see here, again, vacuolization and all happens, okay? So, this one happens, again, vacuoles will be formed, nucleus will be formed, and asymmetrical cell will be formed. Then your vegetative and generative cell will be formed. In that, vegetative cell is the larger cell and generative cell is the Kuti cell, okay? Yes, now all of you got it right. Very good. Okay, so this becomes, pollen grain becomes the gametophyte. This is going to form the male gamete. Clear? Can we move further? Yes? Microsporogenesis is pakka clear, no? So, Monica, you understood the order, the question that you had asked. You start from sporogenous tissue, then you go to the your PMC or MMC. MMC undergoes microspore tetrad. Microspore tetrad forms your male gametophyte. Male gametophyte later gives to the male gamete. Okay. Tapetum. Listen dear, don't get it confused. Tapetum is giving the nourishment for the growing entire, what? Your sporogenous tissue. that sporogenous tissue has to undergo division to form the pollen grains. But what is vegetative cell doing? Vegetative cell is giving directly nourishment for the male gamete. Two things are different, right? Here what is happening? This is your microsporangium layers. These cells have to undergo meiosis to form the pollen grains. Pollen grains, again, further vegetative generative is there. Clear to all of you? Yes, Kush, is this clear? Tapetum and vegetative cells are two corner things, okay, south and north pole. Because tapetum is the layer of your microsporangium. Where is microsporangium? That theca I spoke about. That is your microsporangia, right? Inside that, when you go, sporogenous tissue is there. Tapetum is going to provide nourishment for these tissues so that it can divide, undergo division and form and do microsporogenesis, right? What is vegetative cell doing? Vegetative cell is giving nourishment for the male gamete. Clear? One is forming the male gametophyte, one is giving the nourishment to the male gamete. Two different things. Yes, Kush? Clear? Can I move forward? Students, after I teach, please read once again NCERT, then everything will be clear, okay? Because first time you cannot learn this chapter, okay? And if you want one shot of this, let me know in the comments. Okay, ma'am, we want this topic also, we want this topic also. Just put your topics, whichever you want, topics that you need the most, just drop it in the comments. So seeing the topics, I'll be able to take my next lecture up. Now look here, double fertilization. Before I go to double fertilization, I'll give you an idea about what it is, okay? Now students, look here. This is your microspore. Okay, forget about all of that. Your generative cell, okay, generative nucleus will divide by mitosis for what? To form your male gametes. Can you see here? Male gametes will be formed. Now, listen to me carefully. In 60% of the angiosperm, there will be only two cells. One is going to be one vegetative cell, one generative cell. One generative cell will give rise to one male gamete. This is generally what happens. But in some cases, okay, in the rest of the 40% of the angiosperm or 32% of the angiosperm, let's say that what is going to happen in the rest of it, rest angiosperms, okay, again exceptions will be there for everything, okay, rest of the angiosperm students, one vegetative cell and two generative cell will be there. Two generative cell will be there. That means it is going to give rise to what? One male gamete and one more male gamete. Okay, I am writing Mg. Two male gametes it will give rise to. Okay, this is from where your double fertilization comes into place. Clear to all of you? So, in 60% of the flowering plants, it will be one VC and one GC. Okay, one vice president, one general president is not there, no, one VC, vice chancellor and general chancellor, okay. So 60% one VC and one GC will be there, one GC will have one male gamete, it will go and fuse with the egg, process will continue. In some of them, okay, there is a 3-celled stage. What do we call this? 3-celled stage. And in some of them, it is a 2-celled stage. 2-celled stage. Clear to all of you? 2-celled and 3-celled, okay? That is 1 VC, 2 GC, 1 Mg, 1 Mg. Done, done? Clear to all of you? Yes or no? I'll teach you double fertilization, but just understand this concept. Is that clear? 2-celled and 3-celled stage. In 60% angiosperm, it will be 2-cell stage. In the rest of it, it will be 3-cell stage. That means 2 male gametes will be there. In one male gamete will be there. Yes, very good. Syngamy plus triple fusion is your double fertilization. Very good. We'll come into that. Okay. Now, students, this is your female reproductive, right? In female reproductive, we have to learn megasporogenesis. There it was microsporogenesis. Here it is big megasporogenesis. So, the formation of megaspores, the formation of functional megaspores with the help of megaspore mother cell is called as megasporogenesis. Okay. I hope the speed is okay for all of you. Okay. Now, female reproductive system, students, what is there? I told you carpel. Okay, carpel is going to make the unit for your gynecium. Okay, gynecium, carpel is going to make the units. Now, look here, stigma style ovary. Stigma is what? Stigma is the landing place. Okay, it's the landing place for the pollen grains. Okay, for pollen grains. Clear? Pollen grains. Okay, inside the pollen grains, who is there? Male gamete is there. Right, don't forget that concept ever. Okay, stigma, then style is that long slender part, and then inside the ovary, what is there, students? Ovule is there. This ovule is the one who is going to become the seed, right? Fertilization should happen. So ovule is there, locule, placenta. Placenta, locule, you have studied in your 11th morphology of flowering plants. Again, on this channel, we'll be taking that as a revision for your KCET. Okay, now this one ovule, how is that ovule? It's an anatropous ovule. What ovule, ma'am? Anatropous. Anatropous ovule will be given in your NCERT if you see. Students, anatropous means, remember that different types of ovules are there, okay? We have to study anatropous ovule because this is there in almost 80% of the angiosperm have anatropous ovule. What is anatropous ovule? Can you see here? There is a funicle. This is your the micropylar end, and this is your chalazal end. If you look at this, are they in same line? Are they in same line? Like chalazal, let's say chalazal end, micropylar end, and let's say this is the funicle. Okay, too long it became, no? This is, let's say this is, you know, chalazal end. My bracelet is the micropylar end, and this is the funicle. Now this is in a straight line, right? Everything is aligning with each other, straight line. But imagine funicle went that side. Okay, only these two are coming in straight line. That time we can call it anatropous ovule. Okay, just know the name. Anatropous ovule means where funicle, chalazal and micropylar is not coming in a straight line. Are they in a straight line? These two are in a straight line, but this fellow is like, hey, I don't want to be with you people, I will go to one side. So funicle went off like this and ovule became like this. So it's fully 180-degree tilted. Okay, anatropous ovule. Now, we have to know the structure of ovule. So if you look at the ovule, there are integuments, nucellus, funicle, hilum, micropylar, chalazal end. All of this we are going to understand now. Okay, all of this we are going to understand now. So look here, students, this is your ovule. Okay, we have to draw a nice ovule, right? This is like a yes, okay, one second. Yes, can you all see this? Okay, now okay, now students, this is your ovule. Okay, doesn't look like an ovule only. This, okay, let's say this is your ovule. Okay, inside the ovule there is going to be your embryo sac. Okay, inside the ovule embryo sac is going to be there, and around this, okay, around this there is going to be nucellus cells. What cells? Nucellus cells. This is your nucellus cells. Now I am drawing a very, you know, fast diagram so that I do not waste any of your time. Okay, this is your cell. Okay, now look here, students. Now look here, students, please drop it in the comments, okay, because now I will not be able to see all the messages that you send. So definitely drop it in the comments. I am going to check your messages, I mean your comments, okay, whichever chapter you are requesting from me. Now look here, this is your ovule. Now let's try to learn what and all is there in the ovule. First of all, understand this portion is called your chalazal end. What end? Chalazal end. Okay, this portion where the opening is there, this is your micropylar end. Okay, micropylar end. Now, to protect the entire embryo sac, because students, inside the embryo sac, who is there? The queen is there. The egg is inside the embryo sac, right? Like how pollen grains was inside. Okay, how pollen grains was inside. I think another 20 minutes we will be done. Okay, we are not doing the full chapter. We are taking the major 3 topics that has been repeatedly questions have come from the topic. Okay, because full chapter will take us at least, we can do it in a fast like revision 1 hour, 1.5 hours, but if I take it in detail, it will go for like 3-4 hours. Okay, got it? Yes. So students, okay, this is your micropylar, this is your chalazal, and to protect the embryo sac, you have outer integument. Okay, outer integument, and you have inner integument. Okay, inner integument. What are they? They are mainly for protection. Okay, they are mainly for protection. Clear to all of you? Protection is done. Okay, ma'am. Inside this embryo sac, as I told you. Now, students, there is an attachment of the funicle with the ovule, okay, with the integuments, that is called as, I'm so sorry, that is called as the hilum, where in my diagram it is not that visible. Okay, in my diagram it is not that visible, but there is a, you know, like I'll show you the diagram, that you will understand. There is a small portion where the funicle is attaching, which we call it as the hilum. Okay, hilum. This hilum is going to be persistent in your seed. Okay, in the seed also we will be able to see this. Now, this is your embryo sac, but around the embryo sac, what are you seeing? You are seeing a cells called as nucleus cells. Now, this cell is very important for all of you to remember, because an MCQ can come from this. What is the function of nucleus? Function of nucleus is to provide nourishment. To provide nourishment. Nourishment means what, students? Like the food, like how we eat food, no? Same way, to provide nourishment for the growing embryo sac. Clear to all of you? So, embryo sac is a gametophyte or a gamete? Tell me, embryo sac a female gametophyte or gamete? If you answer this correctly, that means you have understood the concept well. If you are answering this wrong, that means you have to learn the concept again. Okay? Tell me, is embryo sac a female gametophyte or gamete? All of you in the chats, quickly. So, nucleus cells are going to provide nourishment for the growing embryo. Inside the embryo sac, students, cells are going to be there, which we are going to see separately again. Okay, which we are going to see separately. Let's do one thing, let's draw it this way only. I'm going to draw your embryo sac here. By then, I want all of your answers in the chat. Okay, quickly answer me in the chat. This is your embryo sac. Okay, this is a fully matured embryo sac. Now, in the embryo sac, what is going to happen? Very good, very good, very good. I'm so proud of all of you. Can put clapping emojis in the chat box for all of you. Okay, very good. You have understood the concept perfectly. Embryo sac is going to be the gametophyte. Inside the embryo sac, egg is going to be there, which is the female gamete. Okay, now I'm showing a completely matured embryo sac. Okay, students, I will teach you Megasporogenesis, but I'm just going to show, or let's do one thing. Let's finish that and come here. Otherwise, some of you might get it confused. Okay, now let's understand what is Megasporogenesis. I told you, formation of megaspores from megaspore mother cell is called as Megasporogenesis. Yes, ma'am, we understood this. So, students, your MMC here also is for what? Is for megaspore mother cell. Okay, megaspore mother cell. So, megaspore mother cell will be there. It will undergo the first meiosis. So, look here, students, many cells are there, no? There is a megaspore mother cell. Okay, the megaspore mother cell, around the megaspore mother cell, you have your nucleus. Okay, they will undergo the first meiosis division and they are going to form a megaspore tetrad. Same thing like microsporogenesis only, no? Something different did you find? No, ma'am, same thing, ma'am. There it was microspore mother cell, here it is megaspore mother cell. Here megaspore mother cell underwent first meiosis division, there also same thing happened. There also a dyad was formed, here also dyad was formed. From here, there is going to be a difference, okay. So far good, no? So, megaspore mother cell underwent meiosis and they formed the megaspore tetrad. Dyad. Okay, megaspore dyad was formed and here the first meiosis happened. Okay, meiosis happened. Clear? Now, next one. See, second scenario happened. Okay, that is from the megaspore dyad. Okay, second meiosis happened. Okay, that led to the formation of megaspore tetrad. Again, do you see any difference from microsporogenesis? No, ma'am, I do not see any difference. There also same thing happened. From dyad, tetrad was formed with the help of meiosis too. Here also same thing is happening. Any doubts so far? Any doubts? Now, the catch here is, even though they formed 4 of them, they formed 4 megaspore, in that only one became functional. What happened, students? Only one became functional. So, look here, out of 4 megaspores, so can you see 4 of them? Out of the 4 megaspore, 3 of them got degenerated. Okay, 3 of them got degenerated. Only one became functional, the functional megaspore. Understood? So we can, when you write the definition also, you can remember like this: formation of functional megaspore from megaspore mother cell is called as megasporogenesis. Now this functional megaspore is again going to undergo mitotic divisions. Okay, so look here, what is going to happen? This is your functional megaspore. Okay, this functional megaspore will undergo the first mitosis division and form what? So degenerate other megaspores, don't worry. Can you see how many cells it is forming? 1 to 2. 2 cells are formed. Clear to all of you? From that, the next mitosis happen, it will form 4 cells. So from 2 celled to 4 celled, from 4 celled to how many cells? 8 celled. So all of you remember why we tell embryo sac it is a 8 celled 7 nucleate 7 cell structure. Can you all say so, students? So look here, from 4 cell stage to 8 cell staged. Yes, very good. Yes, clear? So 8 cell stage we have reached. Okay. Now the catch here is they are all going to scatter. They are all going to run to different place. Right. So once again, can we revise megasporogenesis? So what happens, students? From the dyad. Okay, dyad again. One single megaspore mother cell underwent meiosis, formed the dyad. From there again it went through another meiosis, it formed the tetrad. In the tetrad what happened? Out of the 4, only 1 became functional. The 3 of them got degenerated. Okay, 3 of them got degenerated. That one functional megaspore underwent mitosis, formed a 2 cell stage. Again underwent one more mitosis, formed a 4 cell stage. Again underwent one more mitosis and formed an 8 cell stage. Now students, can I ask you all a question? Are you all ready for one question? Yes or no? Tell me in the chats if all of you are ready for one good question which could have been asked in your, Yes, tell me quickly. Yes. So the question is, how many meiosis and how many mitosis happened for your megasporogenesis? How many meiosis and how many mitosis? I think all of you can answer easily. By then I will draw your embryo sac. Okay, I will draw your embryo sac for all of you to understand the concept easily. Okay. Quickly tell me the answer for it. Answer, students. Yes, it is also called as monosporic development. Why? Because only one functional megaspore is undergoing the rest of the process. So we can also call it as monosporic development. Very good, Babu. Yes, 2 meiosis and 3 mitosis. Very good. Who is that? Vedika. Very, very good. So students, here, what are we supposed to write? We have to write 2-3 terms. That is, it is a monosporic development. Okay. Monosporic development. Why is it a monosporic development? Because only one functional megaspore went through the entire process, and also we have to write that it went through 2 meiosis and 3 mitosis. Okay, yes, correct, very, very good. Okay, very, very good. Okay, next. Now students, that 8 cell stage, what happened to that 8 cell stage is what we are going to see. Look here, this is the 8 cell stage. Okay, 8 celled stage. Can you see 8 cells? Yes ma'am, we can see the 8 cells. From the 8 cell stage, what happened? How did we form the entire embryo sac? So for that you have to know that this 8th cell stage, they all started scattering. Okay, but 3 people, when that said, like when the PD teacher whistle, what happens? We all run, telling that okay, it's free time for us. 3 will go that side and talk about a maths class, 3 will go this side and talk about Hindi class, 2 will come here and be like, you know what, that ma'am was wearing this sari. So everyone will start talking different concepts, right? So same way here also, from the 8th cell stage, here scattering happened and 3 of the cells went to the chalazal end. The cells which are near the chalazal ends are called as your antipodal cells. What cells, students? Your antipodal cells. Okay. Antipodal cells. And 3 of them ran to another corner. Which is another corner called? Sophia. Okay. The other corner is called what, students? It is the micropylar corner. Okay, micropylar end. 3 people ran to the micropylar end. Who are the 3 people? We can together call it the egg apparatus because egg is here. See, center I have drawn one beautiful egg. No, this is your entire egg apparatus. Oh, okay. This is your entire egg apparatus. What does egg apparatus contain? It is going to have egg plus 2 synergids. Okay, egg plus 2 synergids. Now, one thing you have to know about the synergids is, students, that synergids, okay, the synergids have filiform apparatus. Okay, what do they have? Filiform apparatus, finger like, you know, filiform apparatus. This filiform apparatus is going to help the pollen grains, saying that, hey pollen, come here, pollen tube, come this way you have to enter to meet the egg. So filiform apparatus is going to guide the pollen tube to enter inside. So egg apparatus is clear, egg plus synergids. Now 2 people, okay, center of attraction, went and sat off in the center. Who are they? They are together your polar nuclei. Polar nuclei. Clear? So students, we have 8 nucleated but 7 cell stage, right? 7 cell because we are considering polar nuclei as 1 cell. So 7 cell, 8 nucleate embryo sac we have now. Okay. This is a mature embryo sac. Now what is the job? All of you tell me. Yes. Okay, yes. So students, what is the job? All these are your haploids. Okay, haploid. Now what is the job? Here we understood that, ma'am, egg is there. That means egg is going to be what? Egg is my female gamete. My female gamete. Embryo sac is my female gametophyte. Now female gamete and my male gamete has to fuse together. Okay, female gamete and my male gamete has to fuse together. So here what is going to happen, students? As you know, pollen grains is going to fall on the stigma because stigma is the landing place, right? So let's draw one stigma. This is your stigma. This is the style. Okay. All these notes you will get it on the telegram. Okay. Quickly go and join the telegram. This is your stigma, style and ovary. Okay. Now to the stigma, your pollen grains are going to fall. Now it is up to the stigma if they want to accept the pollen grains or not. Okay, let's say this pollen grain got accepted. Okay, let's draw 2-3 pollens. Okay, let's draw 2-3 pollens. Let's say in this one of the pollen got accepted. If this pollen got accepted by the stigma, okay, this is called as pollen-pistil interaction. That means stigma will decide. Now see, when a marriage happens, girl and boy decides, if they want to be with each of them, is their jobs matching, what is their career plan in the future. That means what they are doing a conversation, and in the conversation they are going to reject or accept that person, right? Same thing here also. When the pollen grains falls on the stigma will decide, is this receptive, is it going to be compatible for me? So that is called as pollen-pistil interaction. So it's also pollens and your stigma's compatibility. Who is going to be compatible? Stigma is going to accept. If stigma accepts, it is going to allow the pollen tube formation. It is going to allow what? Pollen tube formation. Clear to all of you? Now, this is your pollen tube germination. Pollen tube germination. So, in the pollen tube, students, let's say the pollen tube has grown, okay, all the way. The pollen tube has grown all the way. if the pollen tube grows all the way down, okay, down all the way like this, okay, same thing goes here. Where is it going to enter? It is going to enter into the embryo sac, okay, because here you have your embryo sac, okay, I am drawing the embryo sac here, and inside the embryo sac who is there? Your antipodals are there, okay, here your synergids, okay, synergids are there, as well as your egg is there, right, egg apparatus, or your entire egg is there, correct, right, this is the embryo sac. So, the pollen tube is going to enter into the egg, clear. Now, by the time, students, as I told you, 3-cell stage and 2-cell stage, no? By the time the pollen tube formation happened, it could be a 2-cell stage. That means one generator cell, one vegetative cell. Let's say it is a 3-cell stage. That means you have 2 male gametes. One I am drawing in blue and one I will draw with dark pink, okay? 2 male gametes are there. Now, you tell me that one male gamete, I know ma'am that one male gamete is going to definitely fuse with whom? One male gamete is going to fuse with the main queen, that is the egg, no? This process is called as syngamy. This is called as what, students? Syngamy. Are you all understanding? I cannot see any of you putting a thumbs up. All of you, are you understanding? Quickly tell me in the chat box, are you all understanding or not? Is there any difficulty that you are facing? Okay, you should tell me, then only I'll understand, no? If you sit silently, how will I understand? Yes. I think, Sophia, I answered your question, right? They are all haploid, okay? Okay, all of you are understanding? Okay, very good, very good, okay? So, students, what is going to happen? One male gamete plus egg, okay? It's going to form your zygote. First, I'll write off zygote, okay? It is going to form your zygote. This is going to happen in 60% of the angiosperm. One male gamete, one egg. Done. Done, done, done. Okay. One male gamete, one egg. This is going to happen in 60% of the angiosperms. You know, this male gamete came from the generative cell, right? The nucleus split and the tube cells, tube nucleus and all formation happened. Now, in this scenario, what has happened? One more male gamete. Okay, one more male gamete, let's say second male gamete, okay, I'll just write second male gamete. The second male gamete is going to directly go and fuse with whom? The polar nuclei, okay, it is going to fuse with the polar nuclei and it is going to form the pen or primary endosperm nucleus, or I can write endosperm, okay, endosperm. Or else you can write P, primary endosperm nucleus. Clear to all of you? Yes? Very good, very good. Yes. So, this is going to happen in the 40%, or we can write rest of them, because again students, exceptions are there, okay? Rest of the angiosperm, this is going to happen. Now, remember that each of the process has a name, okay? Each of the process has a name. What is the name? This process is called as syngamy. This process is called as syngamy, okay? Syngamy, okay? And this one, where 2 male gametes are going to fuse, syngamy is not visible, no? Shall I change the color? In the notes, everything will be visible, but then, you know, here it might not be visible, that's why. Okay, so this one, okay, this one is going to be your syngamy, where one male gamete fuses with the egg and forms your zygote. This one, where the second male gamete is going to fuse with the polar nuclei. Here, what is going to happen? You look at the number and tell me, how is the fusion going to happen? Oh, that's so sweet. Thank you so much. Okay, students, tell me. Can you all think in how the triple fusion would happen? Here, students, see, polar nuclei is what? Your 2n, right? So, this 2n and n is going to form 3n. So, your primary endosperm nucleus will be triploid in condition. So, we call this as triple fusion. Okay, triple fusion. Clear to all of you? Triple fusion. So one syngamy and one triple fusion happened. Now remember this very carefully, that syngamy, okay, syngamy plus triple fusion, syngamy plus triple fusion is equal to double fertilization. Yes or no? Two fertilizations are happening, right? Double fertilization. Okay, double fertilization. So this is the formula that all my students have to remember. What is it? Syngamy plus triple fusion is equal to double fertilization. Any doubts in this so far? Any doubts in this so far? Anything that you have, please ask me. So here you can see the tube nucleus. Okay, here the tube nucleus. Your generative cell nucleus is going to split. Okay. Yes, double fertilization is double clear, double triple okay, yes or no? Double triple okay to all of you? Microsporogenesis, Megasporogenesis is clear. Now I'll show you an image since you understood. Okay, so look here students, this is your pollen grains. Okay, you can see that once the stigma accepts the pollen that okay, we both are compatible, okay, we are full dost, okay, compatible, then pollen tube germination will happen. That pollen tube, can you see how they are going and binding with the embryo sac? Because inside the ovule, who is there? Embryo sac is there, right? Then fertilization has to happen. So inside that, your embryo sac will be there. So here you can go and see the fusion. Okay, so here you see this is the pollen tube, this is your gametes, and this gametes are going to enter with the help of whom? Very, very important. The pollen tube is going to enter with the help of your filiform apparatus, because filiform apparatus is going to guide the pollen. Filiform apparatus is going to guide the pollen tube. Yes, going to guide the pollen tube. Okay, one question can come, so be careful. This is clear? Fine. Now look here students, as you can see, pollen tube is there, the tube nucleus and the male gametes are there, male nuclei are there. Again they are coming in, you can see zygote plus, so one male gamete fusing with the egg is going to form your zygote, which is 2n, and one male gamete fusing with the polar nuclei is going to form 3n, that is endosperm. Okay, triploid. Why? Because polar nuclei is 2n. Nucleated, but one cell we consider it, right? So it is going to form endosperm. Now, ma'am, I have a question. Why do we really have to form endosperm? You can ask me this question, no? Ma'am, why do we have to form endosperm? So students, endosperm is going to give, okay, it is going to provide the nourishment for the growing, okay, endosperm is going to give the nourishment for the growing zygote, okay. Endosperm is going to give the nourishment. Clear? Yes? Any doubts? Yes, very good. Guide the pollen, okay. So triple fusion, it is going to be 3n. I have written here, no? Yeah, 3n I have written. Here it is going to be 2n. Zygote is going to be 2n. Normal situation, okay. One male gamete, one female gamete fusing is happening. So students, so far we have understood so many terminologies, right? All of you are clear with this? Any doubts so far? So students, remember in microsporogenesis, you have to remember this much, okay. I am writing a quick, okay, flow chart. I always write flow chart, or we also call it GM Express, okay. What is it? Gopika Ma'am Express it is. So I am striking out this. It is called GM Express, like Chennai Express, okay, Madurai Express, GM Express is going to be there. That's Gopika Ma'am Express. I'll write a quick flow chart for all of you. I'll do one thing, okay, 1 second. I'll write it down by down, okay. So in microsporogenesis, I'm writing the flow chart for all of you. Sporogenous tissue, Notes I will send it as soon as the class gets over, okay? Sporogenous tissue, MMC or your PMC, okay? That will undergo meiosis. Okay, let's write meiosis later to form your microspore. Students, directly you can write microspore tetrad. I am writing here diad, so all of you remember that 2 meiosis is happening, okay? Microspore. Are you all confident in this topic? Will you be able to do well in your exams? Okay, microspore tetrad, each of them potentially becoming the pollen grains. Okay, in the pollen grains you have vegetative cell, exine, intine and all you people can remember, okay? This is the important part, generative cell. And generative cell again undergoes division, tube nuclei will be there, male nuclei will be there, and that leading to the formation of the male gamete. Okay, male gamete. Now we will write some important parts here, okay, like here your first meiosis 1, okay, meiosis 1, here your meiosis 2, okay, meiosis 2, and this is the male gametophyte. Okay, male gametophyte. This is the nourishment, okay, going to provide the nourishment for whom? For the generative cell. Done, 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 clear? This is your microsporogenesis, okay? This is microsporogenesis, this is going to be megasporogenesis. Okay, so here students, same, what is going to happen here? Megasporogenesis, okay, megasporogenesis, what is going to happen? Your MMC is there, megaspore mother cell will form your megaspore. I am directly writing here tetrad, okay, megaspore tetrad, out of which 1 becomes functional. Okay, 1 becomes functional, this is why it's called monosporic development, and 3 will degenerate. Okay, 3 will degenerate, the ones in the chalazal end will degenerate, okay. In that the 1 functional one, okay, the 1 functional one will undergo again mitotic division and it will form 2 cell stage, 4 cell stage and 8 cell stage. And in from the 8 cell stage it will form a fully developed, or I can write mature embryo sac. Okay, mature embryo sac. Can we write some important points here? Like here I will write meiosis 1 and 2 happened, right? Here meiosis 1 and 2 happened. 1 is becoming functional, so this is called as monosporic, okay, development. Okay, monosporic development. Here from 1 cell stage it undergoed mitosis, okay, again mitosis and mitosis. Yes students, clear? Microsporogenesis, megasporogenesis is clear, okay, and then we formed an 8 cell stage, mature embryo sac. Clear to all of you? Done? We are done, okay, we are done with today's topic. And here last, I would request all of you to subscribe to the channel quickly and do let me know in the comments how the session was. Today we discussed microsporogenesis, megasporogenesis, pollen-pistil interaction, double fertilization. It went for a little long because of some technical issues. Okay, so I hope all of you are going to enjoy the session and let me know in the comments if it was helpful. These notes will be shared to all of you on the Telegram channel, so do not worry. All these notes is for you. I am writing it only for you to make your preparation easy. Okay, so are you all happy? Did you all love it? If you loved it, do not fail students. It will take you only like 2-3 seconds to type a thank you or to type, ma'am, I really enjoyed, I understood something that I did not understand. It will take you just 2-3 seconds. It will not take you half of the time I took to make this PPT for you, right? So I want all of you to take that effort because that is your Guru Dakshana for me, and my GMA should always be thankful and they should be grateful for whatever they are getting for free, right? We are not charging you anything. So for this free content, I want all of you to put that one comment for me and let me have a smile, okay, tomorrow morning when I wake up and see this, okay? Yes, thank you so much for joining. Questions will come later, students. We will do KCET question, okay? Right now, we'll try to understand the concept. Tata, bye-bye. Have a nice, good night. Sleep well. And do not forget to comment, share, like video with all your friends and also subscribe to the channel. Tata, bye-bye.